Well, hello.
So our team's looking at some new waypoints, so we're going to just wait a little bit till they get situated to figure out which way we want to go to continue our transects. So we're just going to stand by while they figure that out.
Madison, thanks so much for tuning in. A past SCF was just saying hi. So an SCF is a Science Communication Fellow. That's the role that I have. And our role is to bring all of this awesome science and expeditions to the greater public. So we conduct ship-to-shore interactions with schools and museums. And then we also help to answer the stream of questions that come in. So, hey, Madison. And it's super important to note that the only way that this works is through teamwork. So there's 31 scientists aboard the EV Nautilus and 17 crew. Scientists have backgrounds in everything from engineering, mapping, biology, geology, and the crew, they're in charge of keeping the ship under navigation, helping us to stay well fed. So collectively we're able to do long missions such as this, where this one we're likely going to be on diving for 24 hours. And even though we're not doing much sampling of biological specimens in this dive, we did have some listeners curious how the processing went from our last dive. So Taylor Ann, can you tell us a little bit about how everything went, especially that big sponge that we collected?
Yeah, sure. So we had a lot of bamboo coral that we processed today, a lot of sponges, and sediment cores that we processed as well, and some eDNA water samples. So as far as the bamboo corals and all the other corals that we collected, I'll put them in the fridge first when we took them out from the ROV, gave them their proper labels, and then took them out for measurements and pictures to properly understand like what they are and where they're going and who we're sending them to process. And then after that, we worked on some of the sediment cores. We removed those from the ROV, taped them up, and froze them to be looked at by another lab. And yeah, that's pretty much how it went. It went pretty smoothly. It took a few hours though.
Okay, this is Danny. I've conferred with Jan and Guy back here, and they've told me what they would like to do. So we are headed towards a new waypoint. We have well characterized our soft sediment habitat. So now we are headed up to a new waypoint, and we are looking for some rock habitat. We will be transiting in this mode until we start to see a change in the habitat. At that time, we will do an on-effort transect for 200 meters. And we will go back to flying at about 2 meters or so with a standard angle and heading if we can. So that's our new plan. Okay. Pilots, are we all set for that new plan? No, no, no.
We love hearing from listeners where they're tuning in from, so feel free to put your name, location at the end, and you know, we do our best to say hello and welcome when we can.
Oh yeah, here we are.
Hi guys, thanks for coming up to relieve for the dinner shift. I'll say what I had just said before you got here, because we just started with a new plan. So we are headed to a new point because we characterized the transects that we wanted to characterize the habitat with those transects that we've already done. So we are headed off in this direction to look for some new habitat. So we will be in off effort mode until we get to a change in the habitat type, looking for some rock habitat, at which time we will go to our on effort mode and do a transect for 200 meters at our standard height, speed, and camera angle.
Got it.
Sounds good. Miles, what do you got the ship moving at right now? 25? Okay. Point 0. Not to say that's a little pessimistic.
I mean, we did go backwards briefly.
That's neat. Fun feature. So there's a little line in the water here.
So Miles, how many new points do we have? I can see one on my view.
We made 3 new points.
Okay.
Here's the first one. Second one's hidden in this dark area, right in the valley.
Okay.
And third one's almost at the top.
Oh,
of
it's
the ridge.
Wayne.
Oh, great. Okay, that looks great. Look, good habitat.
Mm-hmm. That looks like the same piece you went by earlier.
Yeah, it does, doesn't it?
But it's not.
Okay.
Wayne.
Taylor Ann, one of our ocean science interns, you just got a shout out from Sergio in Chicago.
Hey, Sergio. Thanks for tuning in again today. I hope you're enjoying the dive.
Greek watch?
Greek? This is the Hawaiian watch. What's Miles still doing here? Do you get dinner relief these days? Well, do you eat at all?
So what you're hearing some chatter about with our watch schedule, we do get relief to take a meal. So if you are on watch, your 4-hour watch shift during dinner or lunch, someone from another shift comes and relieves you so you can chow down and then come back and be productive with a full belly.
I almost prefer relief at like 5:30 instead of 5.
Halfway through your watch?
Yeah, it's more equidistance for me, but if I was 12 to 4, I would show up right at 5.
Yeah.
Oh yeah. If you notice, I come up here like 5 to 5. I'm like, it's not open yet, but you might as well go get a seat.
Claim your potatoes.
Salad's out, you can start there.
We do get incredibly well fed on the EV Nautilus, so pardon us for talking about it all the time.
So part of our exploring down here is that we don't know what we're going to find. So sometimes we have a plan, and then we find that we want to change the plan. So we found what we were looking for early on in that transect and decided we wanted to take a look and see what was off to the left here, which looks like it could have some more interesting habitat, some slope and rock habitat. So that's what we're headed for now. So we are still seeing some really similar habitat to what we have surveyed in the early part of this watch and the early part of this dive, and we're looking for a change in the habitat type that we will then document in this area.
Yeah, I think that's such a great point, Danny. Being adaptable and flexible in research is certainly a requirement. And so we're just responding to what we've seen and what we hope to see.
Some more marine debris there.
Yeah, it looks like it. Let's have a look.
More plastique.
More plastique. Yeah, go ahead, Wayne. Some nicely discolored plastic.
Oh, Ziploc
It's
bag.
a towel.
Yeah.
Oh, no, it is a
Wow,
Ziploc bag.
Ziploc.
It's a good advertisement, isn't it?
Jeez.
Is it?
Not really.
No.
So, you know, plastics were invented in the 1950s, and of course they have a pretty solid purpose, but we've also overused them and created a lot of single-use plastics. So we're using them for an incredibly short period of time, meaning leading to loads of overconsumption. But there's a lot of conversation around our plastics problem. Obviously, we're seeing plastics even
Okay, Wayne,
at
go ahead
these
and come to the wide,
depths.
please. Thank you.
We categorize that as a macroplastic, a larger plastic or a plastic bigger than the size of like a bottle cap or an inch. We also have microplastics that have been found on our ocean seafloor. And microplastics are typically categorized as 5 millimeters in size or smaller.
These ctenophores are gorgeous.
Why are there so many ctenophores here?
We were talking about that a little earlier in the watch, and especially in the Argus view, you can see so many. So we don't really know, but the ctenophores can be influenced by so much the current and the upwelling and the nutrients. So, and we also know that the midwater and pelagic species is really changeable. If you came back and dove here tomorrow, we might not see any ctenophores. We might see
Right.
different species. So that part of the midwater community really fluctuates a lot. So we're not sure why they're here today.
Yeah, this is the most I've ever seen.
Little known fact, all ctenophores are hermaphroditic, meaning they possess both male and female reproductive organs.
Are they capable of parthenogenesis, or they still need to mate?
Eggs and sperm are shed into the water, and after fertilization, an ovoid larva develops called a pseudipid larvae, as I'm reading from earthlife.net.
So, potentially.
Potentially. Free-swimming larvae. There's a lot of different strategies jellies do for reproduction. There's some jellies that go down to the seafloor and lay sacs on the seafloor that eventually hatch out, free float.
What's this guy here? Is that just an anemone sitting on the seafloor? Wayne, you want to zoom?
Yeah, it looks like.
It's deflated.
Deflated anemone?
Yeah.
It's like something took a munch out of it.
Oh, no.
Oh. If you have a bad day.
Maybe it's just sleeping.
It's just giving up.
It looks like a
It's like a pancake.
jelly donut to me.
Ooh, what type of donuts are you eating?
Not good ones.
All right, Wayne, go ahead and come wide, please. Thank you. Thanks.
We might want to enter our jelly observations on jellywatch.org. This is a citizen science project that
They have a
started
really
up.
cute logo.
Yeah, I know the researcher, Stephen Haddock at MBARI started this. He's a researcher at Monterey Bay Aquarium Research Institute, and he started up this jellywatch.org project to engage everybody in helping to monitor jellies.
Huh.
It would be cool if we added some of this.
Definitely.
It looks like
Yeah.
you got some
I think they would.
Got a cliff coming up here.
steeper relief coming up.
You want to do a snap zoom in there, Wayne? And then I'll get ahead. See what's here. Oh, goodbye.
Wow.
Zoom.
How steep is that terrain supposed to be? 20 meter those 10 meter contours there?
Yeah, they're
Those are
tense.
10s.
Alright, great. There's some sponges there.
Looks like you're seeing over the cliff, at least from this.
Yeah.
Distance.
Alright, Wayne, go ahead and come wide.
Okay, so
Start
there
with
is a little bit of rock there. Let's see if we see some more before we start a transect here, and we'll see how steep that looks up ahead. If it looks like we can run a transect up it.
Yeah, it looks like in the sonar we have a target that's pretty hard coming up in Argus, 60 meters out. So it'll probably be where your rock will start.
Great.
So I'll go there faster.
So is this still mostly soft substrate, or have we graduated to mixed? Does it look like there's a little bit of rock mixed in there?
It's still soft substrate. We have just started to see a little bit of rock. So we're gonna keep going ahead. It looks like there's some rocky substrate ahead of us, and at that point we might have a habitat change.
Cool.
How far laid back are we, Miles? There's not much to that anyway, I don't think.
About 70, 75 meters. So stopping now is not really going to help.
No.
But we're not seeing any shadow, so
No.
I think
No.
it's clearable.
I'll just stay as stretched out here as I can. And then
Is that a lipoema? How do you say that? That liponema. Also known as cousin it.
What is it?
I think I saw an anemone called a liponema, which it kind of looks like cousin it. It's just like super hairy, or lots of tentacles all over the place.
5
Take a look.
meters.
I'm gonna look at my guide.
This will be it.
Oh,
There's a little rock.
wow.
Liponema. There it is.
All right, I'm starting to come up with it.
Oh, okay.
Brevicorn. Cool.
Does this look like it goes for a while, or is we're gonna come to the top of it?
We'll see pretty soon.
Should only be maybe 30 meters tall.
Yeah, it looks like we'll come to the top.
Argus
Starting
is at
to-
the top here now.
Well, this is pretty
Wow.
interesting rock structure. I want to slow down a little bit if we can.
past
We
a
can't. We
coral.
can come back.
Investigate.
Do you want to come back and investigate this?
Yeah, possibly. Can you drop a target here?
Oh, there's an octopus.
Oh, was that under the ledge?
Yeah.
And then we're at the top.
All right. Ship's starting its way back. Well,
All right, Jan and Guy, if you're in the lounge, you want to let me know what you want to do here. We can go back and investigate that, or I can keep moving to the next waypoint.
Maybe go ahead and zoom in, please.
That wasn't a very long feature, but it was pretty interesting.
This is cool.
Beautiful.
Any little thing that sticks out, like a rock or a ridge or anything, and it's just like all kinds of life clings to it.
Yeah.
Yep.
Nice.
All right, so would it be, we kind of zoomed up that wall pretty quickly, but it was pretty interesting. Is it possible to go back and investigate that?
we're in the process of that.
Okay.
All right, Wayne, go ahead.
That was a
Come
nice zoom.
Do we know what kind of crab that is?
I don't know. We have some primnoid corals there. I'm going to chat with Guy for a sec.
Could be an Oregon hair crab. That's one
Yeah.
option.
We were talking about like the Paralomus genus, but we didn't know which species it was.
Huh. Interesting.
You just want to hang out and sort of
Yeah.
loop around.
See if we can.
Paralemus
putz around.
multispina.
Just means we have to find
Potential
something
for
to look
that.
No, yeah,
at.
you were saying.
Something to look at.
Hair
Yeah.
crab.
How much he sent her up on ROV on the, yeah. Thanks.
A viewer would like to know if we've been seeing any pom-pom anemones, or are we mostly seeing still the fly traps?
We did see one pom-pom anemone, the liponema, and they are the ones that are in the seafloor and just really hairy. That's why I call them cousin it. But these are not pom-pom anemones.
Okay.
Oh, we just take a look at a fish for a minute while we wait for the ship.
Follow that fish.
Yep,
Follow
there's
the
that
fish.
flat nose. Last time we had a
You
flat
buddy.
nose that did like a spin move for the camera.
Super cool.
Let me just put a laser on his eye.
Just photobomb.
The sorting hat.
I know. What the heck? Gryffindor, we already said so. I leap. Wayne, you want to come full wide, please? Oh, sure. Yeah, go ahead. Yeah. Yeah, sure. Oh, you're good. We're kind of in a standby mode, so if you guys see something you want to look at. Maybe this guy, photobomb. Yeah, I'm dragging in the dirt there. Give myself a wag here. Okay.
Strawberry ice cream.
Yeah, man.
Huh.
Yep. Till tomorrow, Jess. No. She'll be here. All right, we're heading south.
Heading south? Looks like
Heading
we're heading
south.
north.
Well, you're pointing north.
I'm pointing north, heading
We're bearing
south.
south. Is that better?
Oh, not this again. So, just as everybody is a witness, Jess did that, not me. Why? Why is it in the mud? Z bias, minus 36.
That's good.
Really?
It's nice and still.
Are we on the transect?
You know, that works both ways because she's going to get in here tomorrow when I go to dinner.
Are we on the transect line right now?
Nope.
No, we're in the mud.
We got deviated from the transect line and came off to investigate, look for some hard substrate, and we found a nice wall feature that we just came to the top of, and we wanted to go back and explore it a little more closely.
Roger.
I'm just going to clean myself off here. So we want to head to the south or back up?
We want to go back to the wall we just came up.
Just back up.
Backing up. So this is what usually happens when ROVs go in reverse?
After landing in the mud.
After landing in the mud.
Greetings to our viewers from Georgia, from Virginia, and also from Oregon. Welcome. And Sergio from the Windy City. Who knows Taylor Ann?
Oh, that's not going to go away anytime soon.
Wow.
Hit it with the crops.
And I'll take this moment to say hello to my friends, Leylin and Leydeen from San Francisco. Thanks for watching.
Sorry about the viz, guys. I'm just trying to clean the, there's a big chunk of it there. It's going to be miserable for a minute or miserable for hours. Oh, I think that's the lion's share of it.
If you're just joining us, we are currently in the Greater Farallones National Marine Sanctuary, exploring along 2 transects that were set. Their goal is to characterize the substrate, ideally looking for hard substrate like rock, and looking for corals and sponges.
Sorry.
Someone was crunching some ctenophores.
Frustration.
Squishy.
So we're just settling into this move? Is that the story?
Sorry, say again?
We're just settling into that last ship move and then deciding what's next?
Yeah, rumor is there's a cliff in front of me.
Okay.
Judging by my altitude when I backed up there, should be 10 meters dead ahead.
Okay.
But no, not really seeing it.
Yeah, we had headed off on this heading and come across a wall. It wasn't very tall, so we kind of came up it kind of quickly, and we wanted to go back and explore it because it looked like there was some interesting corals and sponges on it.
Okay.
Dan, Argus is getting pulled around.
Roger. I'm going to come back to the south a bit here.
Okay.
It's my turn. I was wondering, why is it not turning? Because my leash is tight.
Hmm? Oh, yeah. I'm coming back. Slowly. There you are.
Oh, it's just miserable.
What the heck?
I'm going to come down to the bottom, guys, and we'll come up, and we won't have this dust trail. And
Science team, we've got a viewer asking, why even at these depths is the water so blue?
Looks like they're just getting settled, Dave. We might have to repeat that.
Oh,
Oh yes, we switched out for
thank
a dinner
you.
shift, so yes, please repeat. Do you hear him?
Oh, crap.
Well, that's pretty. Now that
Okay.
you can see it.
Sorry about that. I was trying to explain that the water itself doesn't, in and of itself doesn't have a color, but it's the lighting that we use and the reflection and the absorption properties of the water that make it appear to be either greener or bluer. So.
okay,
Great,
we're at
that's
the
an
bottom
excellent
of the cliff.
explanation. Thank you, Wayne.
Do you want to go east, west, or up or down, or
I was given instruction to follow it up, do some
follow it up?
exploring.
Roger. I think we have to move the ship.
All right, so do you guys want to kind of go that way and up?
Oh, let me look. Yes, let me look at HiPAC.
Let me zoom out a little bit.
It looks like the cliff is pretty much to our north. There.
Have you already gone to that point that says Guy 1? Or no?
I think the ship went that high. These are the Argus and Herc trails, so they only went up to there.
Okay.
And now they're about down here.
Okay.
So we can kind of go that way.
Yeah, let's do that. We could do like a zigzag along this.
Okay.
Up, down along
All right.
this feature.
Yep. So getting you guys that way. How about a 027 move? Step it up. All right.
Please.
Bridge nav. Can I have a 10-meter step bearing 027? Thank you.
Let's go 20.
20? All right. Bridge nav. Can we make that a 20-meter step, please? Thank you.
It's looking at the distance there. So I think she's 40 meters away from the cliff top.
Can we have lasers on, Herc?
Lasers. Where are they?
Commander lights. Scale laser.
Thank you.
Welcome.
This is nice to see some rock habitat.
See if I can, you moved the ship. The bearing was
027.
Roger.
Are we waiting on a ship move?
Yes, we are.
Can I request that you go down a bit? I saw black coral that I'm interested in. Is that
Roger.
Thank you. So it's, let's see, how do I tell you where it is?
You can come point with a stick if you want.
Okay, so it just popped into the bottom left hand of the screen.
Bottom left.
It's a pinkish color.
I see it. Just parallel with the lasers now?
Oh no, it's now out of view.
Oh.
So if you came up a bit and then went to your left.
Coming up.
Along that crack there?
So it's even more to the left if the vehicle can move to the left.
Okay.
I guess when you adjust the...
There it is.
I see it now.
Left corner.
I guess when you adjust the auto XY to 1 degree, it also does auto heading.
All right, so now it's in line with the lasers. Can we zoom, please?
Yeah, go ahead. Go ahead.
So this is a type of black coral. It's a little bit different than the ones that we'd seen, the bathypathies. But I believe they might have collected one that looked like this on a previous dive. Can we get any more zoom, or is it hard to hold position?
No, I can get back down. Ah. I'm gonna turn on some autos here and mess with the controls. They're not behaving.
Looks like it has something growing on it.
Oh, yeah.
Or hydroids, maybe.
The green.
Yeah, the brown, brown green.
Are you Lippypath?
It might be a lilipathys, yeah.
Can you zoom out just a bit, Wayne? Let me see if I can.
Stand by.
Or it might be this other one.
Thank you.
That
That's
one.
good there.
Paraffin. These. Are you guys in the mood to collect anything?
We are always in the mood for collection. It's what we do.
The ship's not going anywhere if you want to settle in.
Okay, coming wide.
What a beautiful rock.
Yeah.
I'm going to cheat this time and use the
scoop?
high definition camera to land the vehicle. We usually do it with the bubble cam and you guys don't see it, but it's struggling at the moment.
always interesting to see Hercules taking a sample. You get to see its manipulator arms in action and all of the fine movements that they can make. So Hercules has 2 manipulator arms, which are able to take biological samples and rock samples, but also has the capacity to take water samples as well as cores, sediment cores.
Would you like a partial zoom?
Yes, please.
If it's possible, I'd like the entire individual since it's not too large. If you cut it close to the base so we get multiple branches, that would be great.
Can do. You want to zoom right in once, Wayne? See if this,
Sure.
if I can see an optimal grab spot there. Can't really make out where his base is.
No, I'm good. Yeah.
You're going to avoid the ice cream? Not going to avoid the ice cream?
I am avoiding the
Where do
ice
you want
cream.
to put this?
Wow, that is willpower.
And the sample's going to go.
We have all the bio boxes available. Did you guys want to put it in a larger or smaller box?
Whatever fits best.
Probably larger. Do you think this is a sinker or a floater? I should ask that question before.
Yes, it looks like it does have some floating potential, but I'm hopeful it's not as floaty as a sponge. Is that helpful?
Yeah, that's helpful.
Not as sinky as a bamboo coral? I don't really want to slurp it if that's what you're...
No, I was just trying to, I don't think we get it in one of the small bio boxes, and I can't remember if the front porch is smaller than the big boxes on the
Oh.
I was just trying to work out which one was smaller, because it could go in the next biggest one, which I think is the front.
I don't know. Not a data logger. Hey, data.
Yeah, they seem to be similar in size. I mean, the chart shows them as the same size here, and I only saw them once in person.
You want to push the porch out?
Yeah, I'd say.
Roger.
We'll go for the low-hanging fruit there. So it's,
yeah.
If it hits the cliff, stop.
I might 1 second.
You're at full wide.
Roger, thank you.
Oops.
You said it was sample 24 data?
Yes,
Okay,
sample 24, 024.
Nice shot.
So
Our pleasure.
And one last request, since we are in an area with other corals, can we grab a Niskin?
Sure. You want to pop it right here while we're...
Maybe could we pull away from the wall a little bit? And I don't know if that
Science one.
helps get other coral eDNA in the water.
Sure, yeah.
Because there was a bamboo coral
That's not
right
her.
below us.
And can you remind viewers why we take eDNA near some of our coral samples?
Sure. So just like we as humans, you know, slough off skin cells, corals have cells that they have come off of them into the water column. And so these can be collected in water samples. And there's a researcher with the
Should be good for
National Marine
AutoXY,
Fisheries
yeah.
Service
Should be
who
good
is
for
working
AutoXY.
on
Yeah, I think so.
determining the
I won't touch the button.
genomics of corals from water samples. So with the end goal of having these coral signatures mapped from water samples, then in the future we can better understand coral populations and areas without having to actually collect them or survey with ROVs or nets.
Thank you. I just find it to be such a cool way to learn about our environment, and I know it's more of a kind of emerging science as to what details it tells us about an area, but obviously learning about living organisms through taking water samples and sequencing DNA is really exciting.
Yeah,
Can't wait to hear more.
These water samples are also used by another researcher for carbonate chemistry analyses. So that helps her understand what kind of environment the corals are living in terms of carbon saturation, and that gets down to the bigger questions of ocean acidification. Well, I'm going to hand it back over to Danny. Thank you for the collection, you guys.
Thanks, Caitlin.
See ya. Caitlin, you just get to come and do all the fun stuff.
But it's just a great reminder that all of these different collections help to tell a clear story, and that comprehensively they teach us a lot more about our environment. So that's why we collect often a lot of these samples in the same area to get the clearest picture possible.
Can you turn on the porch light for me?
Yeah.
I've got it now.
Sorry.
I didn't see it trip, did you? Oh, yeah.
Mm-hmm.
Something.
Oh, the one on the end.
Yeah.
Yeah. Good job.
Okay.
What was that sample number, Data?
What the?
7.
That was 024.
Okay, thank you.
Or 0225.
Okay.
Yeah.
I don't have the porch
025.
light on my...
No, it's down LED, I think.
No, look, you have 7 and I have 6.
Really?
Yeah, that's so weird.
That is weird.
Well,
I guess you can't turn on the porch light.
I can't.
Just to clarify again, that was sample 025, the Niskin after the collection of the black coral, which was 024.
All right, I'll double check that. Yep, looks good.
All right, so Danny and Jenny are back on the scientist's row. Looks like I've got a nice sample there, that black coral, and I don't know how well this feature has been explored, but we'd like to check it out a little bit.
All right, 1 second, we're just gonna switch back to Miles and then he can get you going again.
If you see
Roger.
If you see Greg down there, could you ask him to come up for a minute? Maybe Miles knows. What's this one? Encoder. Yeah, encoder. So it's really weird. When I put it in auto heading, I can't change the heading. It's like it's auto heading lock.
Is that a new feature?
Yeah, and it's something maybe since the XY.
All right, do we have a information about how tall this feature is? Can we see that from our sonar?
Not yet. I don't know if anyone got a depth at the bottom. I can come back down, and then once we get to the top, we can compare the notes.
That's all right.
Or I can just ping the altitude. So we're 5 meters up. I
Okay.
think it's about 11 to 12 meters.
Okay, that's great. So what we'd like to do is kind of go along this feature to the left and see how much it extends, and see if it wraps around with this contour, and if it does, we would follow that along up to our next, which I think would lead us up around to our next waypoint. Let me see, zoom out and see if I'm explaining it correctly. Rather than going up and over the top of this to the next waypoint, to see if we can follow this contour around if this feature extends that way. So
around being to the west?
To the west, yes.
Roger.
So we're seeing some black corals here. These are bathypathes. There's one right to the right of the screen, which looks pretty healthy, but there's also one coming up where the lasers are, which looks like a little unhealthy, a little beaten up. Can I get a zoom on the bamboo coral that we just passed? It's coming to the right of our screen, and then there's a black coral below it, just to the below that coral and to the left. So first on the bamboo.
This guy here.
Yep, that's right.
You ready?
Yes, sir.
Okay, zooming in.
And Danny, one of our viewers was curious why the name bamboo